Hello, everyone. So today we're going to continue the discussion we started related to genetic and developmental disorders. Last time I saw you, we started looking into the types of inheritance, the Mendelian pattern, and we've introduced terms such as homozygous versus heterozygous. We said every time we have two identical copies of a particular gene, so let's say this gene or this trait versus this trait, because they match, we call that, in genetics anyways, we call that homozygous. Heterozygous is when an individual inherits a single copy and they will end up with non-matching copies. So if we say an individual has a trait with this genetic makeup, then that will make them basically a heterozygous. Dominant versus recessive. A dominant trait is a trait where you need basically one copy for the problem to manifest. So if, let's say, this is the disease or the capital B is responsible for the disease, both of the examples I wrote would actually manifest or express that disease. With recessive, on the other hand, we need two copies of the gene, the actual recessive gene, in order for the disease or the condition to be expressed. So based on that, we said diseases can be classified into autosomal versus sex-linked disorders. There are dominant autosomal disorders, recessive autosomal disorders, dominant sex-linked disorders, and recessive sex-linked disorders. Most of what we'll focus on in our discussion will be the autosomal type of inheritance, both dominant and recessive. We also have a category we'll conclude a discussion with, which is the mitochondrial gene disorder. If you recall from biology, the mitochondria has its own DNA and it gets passed from our moms. So within that DNA, there are a handful of genes that will basically carry certain information. So every time there is a mutation, there is a problem within that genetic material, within that DNA, then we'll see a disease process of some sort. So let's start with the first category, the autosomal dominant. We said by definition, we inherit those through autosomes, and that's basically most of the chromosomes that we have. And this would involve at least one dominant allele, which will result in the expression of a trait. All right, so remember, it's this or even this. As long as we have one, excuse me, as long as we have one copy of that dominant allele, then basically the disease or the trait will be expressed. This is what you see in here in genetics is something we call a pedigree. A pedigree is kind of a tree representation of how mutations get passed from one generation to another. So I'll tell you some points about the convention of a pedigree. So usually we go top to bottom in a pedigree, meaning that what you see here will be the first generation. And of course, first generation could mean anything. It could mean your parents if we're starting the pedigree at that level. It could be our grandparents if we start the pedigree at that level. And then this will be the second generation. All right, in the first generation, you can see there is a square and a circle. The square is usually a representation for male, so this will be the male, and the circle is a representation of a female. All right, then you see the color coding. Usually if you don't have colors, the shaded area, so a shaded box or a shaded circle, means that they have it. An empty circle, an empty box, it means they don't have it. And a half shaded circle or square means they are carriers. All right, so those are kind of the rules, if you wish. All right, now if you see one line, so in generation 1, you see one line connecting the two, it means that there is marriage. All right, if instead of seeing one line, you see two lines between, let's say, a male and a female, the two lines represent consanguinity. That's when, for example, people who are related to one another basically marry and reproduce children. So what you're seeing here, the affected male and the affected female are depicted in the vibrant yellow and vibrant red, or dark red, dark yellow if you wish. The unaffected will be the light yellow and the light red. So in our first example, in pedigree A, the first generation, we see an affected male, so a male who has the disease, marrying a female who doesn't have the disease, and then they have a bunch of kids. So let's number those kids. Kid 1, 2, 3, 4, 5, 6, 7, and 8. All right, can you guess who is what? Well, kid 1, the daughter is fine. She's not a carrier, not affected. Kid 2, well, that's a boy, and he doesn't have the disease. Kid 3 is a girl. She doesn't have the disease. Kid 4, it's a girl, and she has the disease. 5, it's a boy, doesn't have the disease. 6, a girl that has the disease. And then 7 and 8 are boys who have the disease. In pedigree 2, again, first generation, second generation. First generation, we're seeing a male who has the disease, or the mutation if you wish, marries a female who has the disease, or the mutation. And again, they have a bunch of kids, 1, 2, 3, 4, 5, 6, 7, and 8. Now, when both parents have it, they will pass it into more kids. So the odds of expressing it in their children will become higher. And that's what we're seeing. Now, out of the 8 children, only the daughter, the first daughter, and the 5th son did not have the disease. Everyone else has the disease. All right? Because now, when the mom has it and the dad has it, each one will give a copy to their kids. So we'll see more kids having that disease, and so on. What are some examples of diseases that are carried through this pattern of inheritance? Well, it could be Marfan syndrome. Marfan syndrome is a genetic disease. There are multiple types of Marfan syndrome, all of which affect connective tissues, particularly things like collagen. We also have genetic diseases that affect bones. This would include osteogenesis imperfecta. That name tells you a lot about what the disease is. And achondroplastic dwarfism, which is one type or one form of dwarfism. Other examples, so when you think of cardiovascular system, the one example I have for you is something we'll discuss in more detail in the cardiovascular section. It's the hypercholesterolemia. Hyper meaning more or high levels of cholesterol in the blood. Kidney disorders would include things like adult polycystic kidney disease, something we'll talk about in the renal system, and Wilms tumor, which is a kidney cancer, usually affects children. In the blood, we have spherocytosis, which is a genetic problem where there is an anomaly within the red cell wall that we end up with basically spheres, or red blood cells that will look like a circle as opposed to a disc. And then in the nervous system, things like Huntington's disease, we'll discuss that in more detail, and neurofibromatosis. And in the GI tract, things like familial polyposis coli, which is a condition that could predispose to bowel cancer. All right. The second example is the autosomal recessive diseases. Now, again, autosomal, it means the mutation is located on the autosome. The inheritance would involve 2 recessive alleles in order for the condition or the trait to be expressed. All right? If we have 1 recessive allele, so only this will have the disease. Someone who is genetically this, it means they have 1 copy of that mutation. It means they become a carrier. All right? Now, if they marry someone who is a carrier, so let's say this is the woman, she marries this man, eventually if we do the cross, one child may get the little a from the dad, the little a from the mom, and then they produce a child who has that disease. All right, so here what we're seeing differently compared to the other pedigree we've seen is the carrier state, all right, the half-crossed circle or the half-crossed square. So again, we have generation 1, generation 2, and then generation 3, which is depicted in 2 areas. So a carrier dad married a normal mom or unaffected mom. They have 2 kids. They have a boy who doesn't have the disease, unaffected, and a girl who is a carrier. All right. The boy marries an unaffected woman, and then they will have normal kids because both of them are unaffected. The affected woman here, or the carrier woman, will marry a carrier man, and then that will result in, again, look at the carrier, 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 affected, affected, and then normal. All right, what are some examples? Usually with autosomal recessive disorders, the convention or the theme is that it involves mutations where you see deficiency in something. Deficiency in a receptor, deficiency in an enzyme, a protein of some sort. So here are some examples. Cystic fibrosis, that's one of the common things you probably see in the future. Different forms of anemia like sickle cell anemia or thalassemia. Tay-Sachs disease, which involves an enzyme deficiency. Niemann-Pick disease. Hurler's, Hunter's syndrome, phenylketonuria, and albinism. Most of these, just so you know, they involve a deficiency of something, a protein of some sort. All right. The other examples would include the sex-linked mutations here. They are passed on a sex chromosome, and most of these disorders are linked to the X chromosomes. All right, and you see that males are more likely to be affected. Why is that? Well, let's try to figure it out together. If it's an X chromosome, remember, the female, a male and a female. Genetically speaking, a male has an X and a Y chromosome. The female has 2 X chromosomes. If there is a mutation in one of the X chromosomes, so let's put an X here. All right, let's say the blue is the affected or mutated chromosome. Now, when you look at the male has 1 copy of that X chromosome. So if something goes wrong with that copy, then they will have the disease. Female, on the other hand, she has 2 copies of the X chromosome. So even if there is 1 mutated X, there is another normal X. So they may not manifest the disease. In fact, it's fairly uncommon to see sex-linked, X-linked to be more specific, X-linked genetic disorders in females. That's why you're seeing that they are more likely to be seen in male patients. All right, so here we're again generation 1, generation 2. We have a male carrier, and we have a normal or unaffected female. All right, the male will pass on the mutation to his daughters, because remember, the daughters have 2 X's. 1 X will be derived from the mom, 1 X will be derived from the dad. All right, so if she gets 1 copy, she will be affected. If that daughter marries someone, regardless of their genetic makeup, then she will give one of her X chromosomes to her son. So with sex-linked diseases, they basically, females are carriers, just like what you're seeing here, and males are affected for the most part. The last of these disorders is the mitochondrial gene disorder. These are related usually to energy production, the mitochondria is, and these diseases are only maternal, so they only come from the mom. Why? Because remember, our mitochondria are derived from our moms. All right? When the egg cell and the sperm cell unite, only the mitochondria and the egg cell will continue the journey. No mitochondria from the sperm cell will actually be passed into the zygote. So that's why all the mitochondria you have came from your mom, all the mitochondria your mom has came from her mom, and so on and so forth. So it's never passed by men. Now, more relevant probably for your scope is the polygenic disorders, or inheritance that would apply to polygenic disorders. What does that really mean, polygenic disorders? Well, basically, this is a type of genetic disorders where we see involvement of multiple alleles. All right, they could be on different chromosomes, different locations, and we see a phenotype change, an expression based on the mutation that gets passed on. This is a fairly complex form of interaction between multiple factors. It could be environmental factors. So because of the complexity of what could result in a problem, it becomes really hard to predict what sort of issues can we see. Some possible example would include things like Turner syndrome and Down syndrome. The Turner syndrome is when a female is born with one X chromosome. So she loses one of her X chromosomes. This is a 0, so there is really no X, just one. And Down syndrome is when someone inherits a third copy, so have 3 copies of chromosome 21. All right, remember, under normal circumstances, we have 2 copies of each chromosome. So for 21, chromosome number 21, we have 1 copy from our mom, 1 copy from our dad. All right, but if we accidentally get an additional copy, then that could result in the trisomy, so extra copy. Monosomy is minus a copy. A lot of these diseases can be investigated by a karyotype, so any chromosomal alteration. All right, here the term karyotype is really kind of the mapping of chromosomes. All right, so you see the 2 copies of chromosome 1, 2, 3, all the way to 20, 21, 22, and then there is an X and there is a Y. So this particular karyotype is from a genetic male. How do I know? Because there is an X and a Y chromosome. If there were the 2 X's, like this, then it would have been a female individual. All right. Now, these chromosomal alterations, so whether getting an extra copy or lose a copy, usually result from cell division, particularly meiosis. All right, remember, meiosis is a type of cell division that affects gametes, sperm, egg cells. So the term non-disjunction is really failure of chromosomes to separate. So what would that look like? Well, when you look at, for example, how chromosomes separate, remember, the goal of meiosis is to reduce genetic information, because the egg cell and the sperm cell, they have half the genetic material. So they have 22 autosome plus 1 sex chromosome. And then when an egg cell binds with a sperm cell, and 1 sex chromosome. So then when they bind, we'll end up with 44 autosomes plus 2 sex chromosomes. So we have to kind of divide or reduce the genetic information by half in these egg and sperm cells. So when you see during the first meiotic division, we divide the chromosomes into 2 cells. By the second meiotic division, each cell will get 1 copy. So this is, I'll put an N here for normal. This is a normal gamete. This is a normal gamete. Here, those 2 chromosomes, they fail to separate. So both of them ended up going to one gamete, a sperm or an egg, and then the other gamete is empty, did not receive any genetic information. All right, let's, for the argument or for the purpose of discussing this, let's say this is an egg. All right, and this is an egg. Now, let's assume that this egg is going to be fertilized by a sperm. All right, so an empty egg with no genetic material gets fertilized by a sperm, so plus a sperm, which will bring the 22 autosomes and the 1 sex chromosome. It will result in half the normal genetic information. All right, this one here, it has duplicate amount of genetic information. And now it's going to be fertilized by a sperm. So now it will receive 3 copies as opposed to the normal. So now we have trisomy, 3 copies, as opposed to monosomy, which is half the normal amount of genetic information. This one, the normal, if it binds with a sperm, then 23 chromosomes here plus 23 chromosomes, so we get the duplicate number, the normal amount of chromosomes, or the normal number of chromosomes, if you wish. All right, other terms that we may kind of encounter, well, in terms of what affects our genetic material, well, there are the environmental, the metabolic agents, histones. Histones, they are proteins within the DNA. They could basically undergo chemical reactions like acetylation. There is DNA methylation. The term methylation is adding a methyl group, which will really shut off a particular chemical compound. And then there is the genomic imprinting. We're not going to get into too much detail because this is not a genetics course. I'm just introducing the terms. Now, how can these be assessed? Well, there is what's called prenatal screening and diagnosis and postnatal screening and diagnosis, things that can be done during pregnancy, before birth, prenatal, and after birth, postnatal. And these are usually done to diagnose serious conditions, to provide options for treatment and possible interventions early in life. You know, there are some of these genetic diseases, they have ways to treat them. It would involve some sort of a gene therapy or supplementation with a particular protein, and so on. All right, now with some of these developmental disorders, we'll encounter kind of some of them in future discussions. Things like Huntington's disease, for example, when we discuss the nervous system, and you know, blood disorders, MELAS is something we'll discuss later on, and so on. All right, so the environment, so environmental factors plus multifactorial factors plus any foreign substances that could affect development, we call those teratogens. These can affect genes or chromosomes. All right, if they affect chromosomes, well, we may end up in alteration of the chromosome structure, something we call translocation, or an alteration within the number of chromosomes. So we get half the normal, monosomy, or duplicate, another copy on top of the normal, so a trisomy. At the gene level, again, we could inherit autosomal versus sex-linked. All right, and then there is the mitochondrial type as well. So let's start with Huntington's disease. What is Huntington's disease? It's a progressive degenerative neurological disorders. It's an autosomal dominant, so now we know there are 2 copies are required to actually, I'm sorry, I meant to say 1 copy is required to express the disease. The defect is in the Huntington gene, and that gene is located on chromosome 4. All right, here's a simple mnemonic to help you remember it. When you look at the term or the word Huntington, all right, the first one is hunt. All right, people usually hunt for food, right? So the Huntington gene is on chromosome 4. The primary area in the brain that gets affected by it is the basal ganglia. If you don't know what the basal ganglia is, it's the primary center in the brain responsible for coordination and refining skeletal muscle movement. All right, things like Parkinson's disease is another disease that affects basal ganglia. Huntington is just another one. Now, we see involuntary movements like chorea, dyskinesia. We see cognitive impairment, emotional disturbances, a wide spectrum of problems. In terms of diagnosis, usually it requires medical history, so family history, genetic history, physical exam for all the brain-related or neurological symptoms, and then evaluation of the emotional, the cognitive, the mental status, genetic testing, blood testing, and then brain scans, various forms of scans. CT, computed tomography, magnetic resonance imaging, MRI, and positron tomography, which is the PET scan. There's really no cure for Huntington's disease. There are some drugs that could help skeletal muscle movement, as well as all the psychiatric complaints for the patient, and a lot of supportive care. Genetic screening is available. In fact, a lot of these commercial genetic tests that you can do these days, like 23andMe, these actually can tell if the individual is a carrier for a Huntington gene mutation. And that's, of course, where a lot of individuals would refuse to do these tests because they don't want to know if they are carriers, because the quality of their life would change significantly if they know that they will develop a very progressive neurological disorder by age 50, and they'll be probably dead by age 55 to 60. All right. The other example on our agenda is sickle cell disease. This is a blood disorder. It's a single gene mutation, and it's an autosomal recessive. So that means we need 1 copy, 2 copies. I'm getting those wrong tonight. So we need 2 copies of the recessive gene in order for the pathology or the trait to appear. All right. The heterozygous, so this would be a carrier. Homozygous, small s's, will be the actual disease. It's a type of point mutation which will alter the protein structure. Which protein? We're talking about the beta chain of the hemoglobin protein. So this will result in an abnormal shape red blood cells that they will look like a sickle. All right, so they change shape, they become abnormal, and when they are abnormal, they could potentially stack and occlude small blood vessels, and that could result in basically deprivation of blood flow into certain tissues and organs. So what can we see? Well, we see jaundice because a lot of these abnormal cells have to die in the liver. So a lot of hemoglobin gets generated, a lot of bilirubin gets generated. We see the yellowish discoloration. We see anemia because, remember, red cells, they carry oxygen. When they're abnormal and dying, well, who's going to carry that oxygen? We see pain and organ damage. Those are because of the occlusion of blood vessels. When you deny certain blood vessels from blood flow, we see pain, we see damage in organs. the risk of infection will go up, and of course the severity of the symptoms will be related to the percentage of the abnormal hemoglobin in the circulation. So even the disease itself is kind of along a spectrum: severe, mild, and moderate. Again, it may require genetic testing or lab-based analyses, either of hemoglobin, determining which type or the proportion of the abnormal hemoglobin, complete blood count, analysis of urine, and tests for liver and kidney function. As far as treatment is usually directed towards symptoms, so symptoms such as pain, symptoms such as the jaundice, avoidance of temperature extremes, particularly cold, because remember, cold results in vasoconstriction, so that could result in decreased blood flow to the peripheries. And prevention and treatment of infection, and reduction of any form of stress. The one interesting thing about sickle cell disease, for those of you who don't know, is that the sickle cell, so this abnormal cell, becomes actually resistant to infection with malaria. All right, malaria, the parasite, has the capacity to infect normal blood cells, but not sickle cell disease. All right, so that's, like I said, that's kind of an interesting point, because this is kind of a sort of discussion that will show, you know, how evolution works. It's not about how good or bad things are, but about coping with the circumstances. If you recall, malaria infection is fairly high in Africa, and sickle cell disease is fairly high in individuals of African descent. Why? Well, as I said, because a sickle cell, an abnormal cell, although it's not desired, but it will protect against malaria infection. All right. The next group are the conditions inherited through the mitochondria. The first type I have, or the first example I have for you, is the mitochondrial encephalopathy, lactic acidosis, and stroke. So this is kind of a symptom-based terminology that we're using, abbreviating it into MELAS. So what happens in this? Well, this is a type of mitochondrial gene mutation. We said this is only from the mom's side, and it could result in impairment in energy production and use, either at the level of the nervous tissue or the muscular tissue. So presentation would include seizure, dementia, that's what the encephalopathy is really referring to, lactic acidosis, remember this is when we enter anaerobic pathways, and stroke-like episodes. So really, muscle plus nervous tissue. Other problems we may encounter: hearing loss, nervous tissue; blindness, nervous tissue; migraines, nervous tissue; and vomiting. It requires physical exam to make the diagnosis, lab studies, so assessing mitochondrial function, assessing the level and severity of lactic acidosis. Muscle biopsy is also done, and imaging studies to assess the brain. In terms of treatment, it's again based on symptoms. So treatment to minimize the frequency of seizures, and then promote mitochondrial energy production. And of course, you know, if hearing loss is an issue, then a cochlear implant to help with regaining some of that function. All right, the next one on our agenda is Down syndrome. We said Down syndrome is an autosome problem, an autosome number problem. It's an additional copy, so with 3 copies instead of the normal 2, a trisomy of chromosome 21. And now we know that this condition results from a problem with how chromosomes separate, so it's the non-disjunction. Individuals with Down syndrome, they have a wide spectrum of symptoms, some of them related to mental development, brain development, unique facial features, so the slanted eyes, the epicanthal folds, cardiac defects, which usually involves defects in the septa. We'll talk about this in the cardiovascular system. GI malformations, hearing, visual problems, thyroid dysfunction, and the risk for developing certain leukemias, that's white blood cell cancer, would increase in individuals with Down syndrome. Usually, the diagnosis is made based on physical exam and prenatal diagnosis. Prenatal diagnosis will either examine the chromosomes of the baby or physical parameters. All right. The term nuchal translucency is a term used in ultrasound, and then anatomic anomalies can be detected right away, and then of course karyotyping, so visualizing the chromosomes of the fetus to see if there is an additional copy of chromosome 21. How about treatment? Well, there is really nothing that can be done. Early interventions could help with the overall progression of life and the severity of the disease. So physical therapy, speech therapy, occupational therapy. Again, the severity of Down syndrome varies greatly in the population, and it's usually the way to assess it is by looking at IQ. All right. The lower the IQ in some of these patients, the harder it is for them to be independent. The higher the IQ, the easier for these individuals, even with Down syndrome, to be fully independent. May require some counseling here and there, financial, for example, just so that no one take advantage of them, but they could live an independent lifestyle. All right, on the very opposite spectrum, or side of the spectrum, from trisomy, so from duplicating genetic information to cutting it in half, Turner syndrome. So again, it's monosomy of chromosome X. So not autosomes, we're talking sex chromosomes, and we're losing one copy. All right. Certain risks would increase. The risk of diabetes, heart disease would increase. These individuals, again, may have short stature, may have gonadal failure, which could manifest with infertility, delayed puberty, cardiac anomalies, hearing loss, and usually learning difficulties as well. So what are some of the symptoms? Well, you see some examples here, some physical signs anyways. The webbing of the neck, cardiac anomalies. We'll discuss what coarctation of the aorta is later in a different section. Widely spaced nipples. There is the wide carrying angle of the arm. All right, and then decrease in the size of ovaries. They're actually rudimentary, hence the delayed puberty and potentially the infertility. And there is skin manifestations as well. From a diagnosis standpoint, again, we use or rely on prenatal screening test. So we may see swelling in the back of the neck, something is called hygroma. We see edema, short femur, and narrowing in the aortic arch. All these are radiological ultrasound features that could be seen during pregnancy. And then, of course, visualizing the chromosomes, karyotyping, to see if there is a missing copy of that X chromosome. Treatment-wise, again, it's really based on symptoms, so there's nothing specific. Sometimes hormone therapy to kind of trigger puberty, menarche, and pregnancy as well. All right. Now, with sex chromosomes, again, similar to what we've encountered with autosomes, problems could vary greatly. We could receive an extra copy of a sex chromosome, or we could lose a copy. So at the bottom of the slide, you see a couple of examples. Turner syndrome, something we've talked about, is when we lose one copy of X chromosome. Klinefelter syndrome is when we have two copies of X and a Y. So this is really a male, all right? This is also known as testicular feminization, because a male is an XY. The Y has a lot of the traits that males inherit. If we add another X, then the collective effect of these two X's will kind of overrule that Y. That's why patients with Klinefelter syndrome may develop basically high-pitched voice. They are male, but they have high-pitched voice, they have rudimentary testes, and basically, infertile as well. All right, some of the examples we have. So the first one is Fragile X syndrome, which is a sex-linked genetic disorder, and it involves a defect in chromosome X. The mutation is usually with the Fragile X mental retardation protein. It is the leading cause of inheritable, not acquired, mental retardation. And it could affect males mostly, and because it's X chromosome, so remember, if it's damaged, males will have it, females will be carriers. So they may present with distinctive facial features, connective tissue anomalies, below average height, enlarged testes, macro-orchidism, and cognitive impairment. As far as diagnosis, well, it may require genetic screening, prenatal screening, and then treatment is mostly behavioral. So education, because it involves decrease in mental capacity, then they rely on behavioral interventions and so on. All right, the last kind of section is going to be dedicated to neural tube defects. Neural tube defects, they basically are developmental disorders that occur during embryonic development, so really early in development. So usually the defects involve the nervous system, and I have two common examples to share with you. The first one is spina bifida, which exists in 4 different types, and anencephaly. Now, spina bifida is an anomaly in how the vertebrae develop, so that could affect nervous tissue. Anencephaly is when the top part of the skull doesn't develop, so it could result in brain not developing as well. So with spina bifida, it's incomplete closure of the vertebrae. Sometimes it may not involve the herniation or the protrusion of any brain tissue, so no protrusion of meninges, the coverings of the brain, or the spinal cord. We call this spina bifida occulta. It may involve only the meninges protruding or herniating. We call it spina bifida with meningocele. Or it may involve the protrusion of meninges and spinal cord, so nervous tissue. We call this spina bifida with meningomyelocele or myelomeningocele, same thing. Anencephaly is when the skull doesn't close fully and the brain may be exposed with the meninges. In order to diagnose those, prenatal screening is crucial, either done through ultrasound exam, through amniocentesis, that's when amniotic fluid is taken by a needle and then investigated, and usually investigated for markers that could suggest an anomaly, things like alpha-fetoprotein. That's a marker that we rely on to diagnosing a lot of these developmental disorders. How is it treated? Well, it requires supportive care. It may require some surgical correction in the case of spina bifida. All right, so this is what anencephaly would look like. Again, you're seeing that the top part of the head is missing of the skull. All right, a lot of pathology textbooks will describe this as facial, they have frog-like faces. All right, the last kind of topic, which is a low-yield topic, is about the multifactorial inheritance. And as the name suggests, it involves the interplay between multiple factors. Now, traits that usually follow this mode of inheritance would include things like height, intelligence, blood pressure, metabolism, and development. All right, there's so many factors that affect our height. All right, so some people are born too short, some people are born too tall. Now, yes, there could be hormonal influence, genetic component, but at the same time, different factors could play a role. And the same can be said about all the remaining factors that you see listed in the table. With diabetes, although we'll discuss it in more detail in the future, in the endocrine system, it's probably the classical example of multifactorial disease, and about 50% of these individuals have relatives who have diabetes. So that suggests there is some genetic component to it. All right, so that concludes our section. I will see you next week, and we'll start with the next part of our discussion. Thank you.